Okay, so the pH is equal to 6.1 plus the log of HCO3 minus, and that's going to equal 0.014. And then for the CO2, this is the initial amount, which is the 0.0012. So if we do this calculation here, what is the pH based on the open system? Yeah, so in this case here, the pH turns out to be 7.3. Note that this is within that 6.8 to 8.0 range. Okay, so this baking soda CO2 blood buffer system has to be an open system for our blood to be buffered properly. So I'm not quite sure, when you were younger, you got mad at your mom or dad or someone, you held your breath. You said you're going to hold your breath until you died or something like that. Well, see, if you do that, that means now you're trying to make your blood buffer system a closed system. And I guess there's something, I guess you pass out before you die. Is that what happens? You pass out before you die? You guys learned that in physiology or biology? Okay, let's go on to part E. Okay, so your calculation showed that a large decrease in pH part C, a small decrease in pH part D. Based on these calculations, it would appear that HCO3 minus should be quickly depleted in an open system when acid is added. How is the HCO3 minus replenished in blood then? Okay, so in that open system, note that the HCO3 minus is dropped from 0.024 to 0.014. So it's being depleted, or it's decreasing. So how can we get the HCO3 minus back to where it should be? Well, water. Yeah, so based on equilibrium, if we drink water, that should shift the reaction back toward the reactant side, but there is another way here. Yeah, so how do we produce more CO2? Pardon? Yeah, so how is CO2 produced in our body? Yes, I think somebody said it. So how is CO2 produced in our body? This occurs in our cells. Yes, you eat food because when you eat food, the food reacts with oxygen, and that's going to produce ATP, but what else? It produces water and CO2. So once that CO2 is produced, note that the CO2, this CO2 which is produced is going to make more H plus and HCO3 minus, and that's going to raise the pH back to where it should be. Okay. So let's talk about the second blood buffer system. This is the hemoglobin blood buffer system. So this diagram represents a simplified version of the buffering action of hemoglobin. So first we know that hemoglobin is that substance in blood, and what's the function of hemoglobin besides being a blood buffer? It carries oxygen. Note that according to this diagram, there's 4 different forms of hemoglobin. So this is the deoxygenated hemoglobin. And note that hemoglobin, since it's a protein, can either be an acid or a base. Okay, so HGB is the acid form of hemoglobin with no oxygen. HGB is the basic form. HGBO2 is the acid form of the oxygenated form of hemoglobin. HGBO2 is the basic form of the oxygenated form of hemoglobin. So note there's 4 different types of hemoglobin in our blood. The oxygenated form, acid and base. The deoxygenated form, the acid and base. So let's take a look at this reaction here. So when the acid form of deoxygenated hemoglobin reacts with oxygen, it forms the oxygenated form. This is the equilibrium constant for this reaction. Now, actually, let me clarify this one, because that K, that equilibrium constant actually represents the reverse of that reaction. So for this one, if you look at this, HGBO2 to HGB plus O2. So K, we call this KO2 is equal to 1. And then let's take a look at this one right below it. So HGB plus O2 going to HGBO2. The equilibrium constant called K prime for this reaction is 0.032. Okay, now we'll come back to this in just a minute. Okay, so you just told me that when food reacts with oxygen, that produces CO2 and water, and that replenishes the HCO3 minus. And then that HCO3 minus, that's going to produce, in this reaction here, more H2O and CO2. And then we can expel this CO2 by exhaling. The CO2 and H2O, which is formed by eating food, not only produces HCO3 minus, but H plus. And the H plus is going to react with the HGB, the conjugate base of the deoxygenated form of hemoglobin. That's going to reform the HHGB. Okay? Okay, so let's go on to the next slide. Does the conjugate acid of hemoglobin, the HHGB, have a higher, lower, or same affinity for oxygen than its conjugate base, HGB? Okay, so for question A, you want to look at these 2 reactions here and their equilibrium constants. So which one has a higher affinity for oxygen, HGB or HHGB? Okay, so let me just say one thing. Let me go back one slide. Are any of you guys planning to transfer to UC Davis?
Yes.
And are you going to take a major in either biology or biochemistry?
Yes.
Oh, so you'll be taking a class by this guy, Siegel. And he wrote this book, Biochemical Calculations. And he'll cover this in this. See, I got this from his textbook. Okay, so he'll actually cover this in his textbook. He's been teaching for a long time, because note that my version of his textbook from 1976, and I heard he's still teaching UC Davis. So you'll see this again when you transfer to UC Davis and take his biochemistry class. Then when you take these, oh, this is easy. It's all this. Okay, so which form of hemoglobin, the acid form or the basic form, has a higher affinity, in other words, binds oxygen better? Okay, so is it the acid or the base? Why the base? Okay, so remember, based on this, we know that if we write out our equilibrium concentration, that's equal to HHGB times O2 divided by HGB O2. And then for this one, this is equal to HGB times O2 divided by HGB O2. Now note that this equilibrium constant is small, so is there going to be more reactants or more products? We're going to have more reactants. In this case here, we have, it's equal to 1, so is there going to be more reactants or more products? Sorry, more products or more reactants? Equal. Yeah, equal amount.
Equal amount.
So based on these equilibrium constants, which one has a higher affinity to oxygen? The HGB or the HHGB? So who says it should be the HGB? Who says it should be the HHGB? Okay, so how come the rest aren't voting? Okay, so you just told me that based on the equilibrium constant, there's going to be more of reactant, the HGBO2. Based on this equilibrium constant, there's going to be an equal amount of products to reactants. So based on this information here, if there's more HGBO2, that tells us that which one has a higher affinity or binds to oxygen better? Yeah, so the HGB binds to oxygen better, okay, than the HHGB. Okay, let's go into question 2. In the lungs, is the conjugate acid or conjugate base of hemoglobin formed? Okay, so if we go back for one slide, again, is the conjugate acid or conjugate base of hemoglobin formed? Okay, so what's happening in our lungs? Yeah, so the hemoglobin in our lungs, what's happening? So there's oxygen in our lungs, which we inhale. What happens to the oxygen in our lungs? It's going to react with the hemoglobin. Which form of hemoglobin is in our lungs? The HHGB or the HGB? Now, before we answer that question, look at the pKa. So note that the pKa of HGB is 7.7. For HHGBO2, the pKa is 6.2. So we know that the pH of our blood is 7.4. So at pH 7.4, you just said that, okay, so first in our lungs, we should see the deoxygenated form of hemoglobin. Okay, so it's either going to be the HHGB or the HGB form. At pH 7.4, are we going to have more of the acid form or more of the basic form? Okay, so let's take a look at our, are you getting tired of looking at the titration curve? You getting tired of looking at it? No? Then you can draw the titration curve. Does someone want to draw the titration curve of the HGB on the chalkboard so I don't have to do it? I can do it, but I can do it, but then is it helping you if I do it? Okay, so as we know, the hardest part is getting started, so let's get you started on it. Okay, so here's your starts for your titration curve. It's just like exercise, hard part is what happens to it. So from here, okay, we have pH and then volume. Okay, so now based on this here is the deoxygenated hemoglobin a monoprotic, diprotic, or triprotic acid? In other words, how many protons can it donate? Yeah, one. So how many endpoints do we want to show? One. It is one. Okay, so right here is the halfway point. So at the halfway point, the pH is equal to 7.4. No. Okay, 7.7. So at the halfway point, we're going to have how much acid relative to base? Yeah, we'll have half acid, half base. At pH 7.4, which is just below here, we're going to have more acid or more base present? Yeah, more acid present. So that tells us that at pH 7.4, are we going to have more HHGB or more HGB present? Yeah, we'll have more HHGB present. So in our lungs, we're going to have more of this present than that. So we'll say we have more of this present. So